This video is on your lymphatic system. Lymphatic system. And all its associated organs. And your lymphatic system has a lot of functions. The first function is it helps your circulatory system. That's your arteries and your veins. So if we draw your artery, blood will go through it, plasma will go through it, and some will leak out, whether it's through hydrostatic pressure pushing it out, or whether it's oncotic pressure pulling it out. Some fluid will leak out, and your body's smart enough to say, that's not good, I'm gonna reabsorb some of it. But it can't reabsorb all of it, and by the end of the day, you have about 3 liters stuck out in your interstitial space, in your tissue. That's about a gallon of fluid, and we don't want that, right? So your lymphatic system has lymphatic vessels that go alongside your arteries and your veins, and it helps drain that excess fluid back into the heart. Sends it back to the heart. What a nice guy. All right, so that's one way it can help. Another way it can help, and probably a more important way because we're talking about immunology here, is that it helps your immune system. Because your lymphatic system and your lymphatic vessels hold lymph fluid, and that's full of lymphocytes, so that helps out. And also, you have organs associated with your lymphatic system that also have a lot of lymphocytes that also help your immune function. Like your lymph nodes, like your spleen, like your thymus. Don't these things hold a lot of lymphocytes? Don't they have a good immune function? So to help your immune system, and this isn't exhaustive, you have other things like your bone marrow. That's where you make your lymphocytes in the first place. So anything where there's a concentrated amount of lymphocytes, we can say that's associated with our lymphatic system. All right, so let's just talk about the first function. It helps your circulatory system. So it has these little vessels that will drain the blood, and it will eventually coalesce and turn into one big vessel on your left side, one big vessel on your right side. And these vessels will drain into the subclavian, drain back into the heart, help you collect that excess fluid. All right, so the big vessel that everything on the right side kind of collects into is going to be your right lymphatic duct. And that drains your right side, especially the area above your diaphragm. And then the giant left-sided duct that kind of the left side drains into, and eventually back into your heart, it's going to be your thoracic duct, which basically drains everything else, especially your left side. Okay, so these two drain back into your subclavian, drain back to your heart, picks up that excess fluid. Now let's talk about your second function. Sometimes that excess fluid, while it's getting drained back, might hit a lymph node. And when it drains through that lymph node, your lymph node will look at the fluid, make sure there's nothing sneaking in there, no pathogens, no bacteria. All right, so you have lymph nodes in a lot of different places. If you're draining fluid back around here, you might drain into your axillary lymph nodes, where your axillary lymph nodes will look at that fluid, make sure there's nothing sneaking in there. If it's up here, it might be your cervical lymph nodes that do that. But anyways, fluid might go through these lymph nodes, and your lymph nodes will check and make sure there's nothing hiding in there. So let's talk about your lymph nodes. One that they like to test you on, and the one that I want to talk about in particular in this video, is the lymph nodes around your inguinal region, your groin region. You see there are some organs up here that drain into your para-aortic nodes. Para means near, so near your aorta, because these organs are near your aorta. All right. These include things like your kidneys. These include things like your uterus, because these are things near your aorta. But these also include things like your testes and your ovaries. Don't these start in your abdomen? And then they'll descend lower. Your testes will descend all the way. But because they start in your abdomen, they also drain into your para-aortic nodes. So testes, ovaries. Most students don't have a problem with this. They usually remember, oh, this is a trick question. They like to test this on your exam or your step, that your testes and your ovaries, because they start in your abdomen, they drain in your para-aortic lymph nodes. No problems there. Where you might get confused is that the skin around your groin, the outside skin, it's not going to drain in here. Yeah, your outside skin doesn't start from your abdomen and like go outside. No, it's going to drain somewhere else. It's going to drain into your superficial inguinal. What a great name. It's going to drain to your outside area of your inguinal region. right? Your superficial inguinal. So this is your outside skin. This is your scrotum, which is basically like a skin fold. Or if you're a lady, that's your vulva, which is kind of like a skin fold. Or the skin around your anus. So your anal canal below the pectinate. If you remember your anal canal, you have your pectinate line. And below it, that's your outside skin. And then above it, that's your GIT, right? It's your guts. So the outside skin is not going to drain up here. Why would it? It's going to drain on this superficial layer, superficial inguinal. All right, so that's a common way they trick students. They'll talk about a guy that comes in with some sort of scrotal lesion, some sort of scrotal ulcer. And they'll ask you where the lymph drains to. And you're thinking, scrotum, that's your testicles, right? That's got to drain to your peri-aorta. Nope. Your scrotum is not your testicles. Your scrotum is just an outside skin, so it drains into your superficial inguinal. That's a common way students get simple questions wrong, whether it's, they're nervous from taking a step or whatever. Just remember these two things are different. And then lastly, any other internal organs around this area, like your bladder, your prostate, your cervix, will drain into your internal iliacs. Okay. So anything we didn't list before. Okay. There are a lot more lymph nodes, like I said. By your lungs, you have your hilar lymph nodes. Your breast drain into your axillary lymph nodes or something else. And we talk about those in the context of other videos, other blocks. So when we talk about the lungs, we'll talk about those lymph nodes. When we talk about the breast, we'll talk about those lymph nodes. And so it's better to learn those in context of those blocks. So we're not going to talk about them here. All right, you might see in first aid, they'll list all these lymph nodes and you're panicking. You're like, how am I going to remember those? That's too much to remember. Just break it into its respective block. So if you're going to review respiratory, take any respiratory lymph nodes, throw it in that block, and then review it then. So you only have to review a few key facts, and you're not just trying to memorize this giant list. All right, that's the easy way to do it. Don't just try to memorize the giant list. Just throw it into its respective block. All right. We talked a little bit about lymph nodes, but we didn't talk about the anatomy of lymph nodes. Let's talk about the anatomy of lymph nodes. That's what they want you to know. So your lymph nodes is this little nugget, and it has a capsule. It has a capsule over it, so I'll say it's encapsulated. And it also has these little connective tissue partitions, things that break it up into little parts. We call this trabeculated. So it's encapsulated, it's trabeculated, and this is cool because lymph vessels will drain into here. And when fluid goes through, your lymph nodes will inspect it, make sure there's no pathogens in it. And it's kind of cool breaking it up like this. Every vessel gets its own little room, right? Everyone gets its own little responsibility, and so it doesn't have to work as hard. It just says, I'm looking at this fluid right now, making sure there's no pathogens in there. So it breaks it up kind of like this. We call these afferent lymph vessels because they're bringing lymph fluid in. And it will ultimately coalesce into one efferent. And again, that's kind of cool the way to do it. So they all have to go through before they can make it into one efferent. So they all have to be inspected before they can leave. Now your lymph node is broken up into different zones. You have the outer cortex. So there's your cortex. And then the area next to your cortex, we call your paracortex. Remember we said para means next to or nearby. Makes perfect sense. And then the inner area is called your medulla. All right. We'll start from the outside in. Your outside layer is your cortex, right? Your cortex has these little follicles. And these follicles have B cells in them. And sometimes you'll see follicles that are really, really packed with B cells, like really, really condensed, concentrated B cells. They look very dense. We call these primary follicles. These are dense. These are dormant. They're all just kind of concentrated, huddled together, they're just kind of lying in wait. All right, but if there's some sort of infection, they will not be dormant anymore, right? They'll start to rev up. They'll start to rev up, they'll start to enlarge. They'll start to spread out, so they're not so concentrated. There'll be more of them. Because they're not as concentrated, because they're a little bit more spread out, they look pale. They look larger. These are your secondary follicles. These are active. All right, these are active. And they're larger and more spread out microscopically. They're also larger macroscopically. So you get your enlarged lymph nodes. So that's your outside cortex layer. What's your next layer? There, next to your cortex, your paracortex. Absolutely right. So your paracortex has your T cells. Paracortex has your T cells. And these can also enlarge, also make that enlarged lymph nodes. And then finally, your medulla, that's your last inside layer, has both. Has your T cell lymphocytes, has your B cells, which can also become plasma cells and make antibodies. And your medulla will eventually have little holes, have little sinuses that drain all the fluid into your efferent and out your lymph node and send it on its merry way. So that is your lymph node. Let's talk about our next organ, your spleen. Let's talk about your spleen. Your spleen, they want you to know just the general anatomy of your spleen. Where's your spleen located? It's this giant organ on your left upper quadrant. Your left upper quadrant. And they want you to know that it too has a capsule. Capsule. And it too has these little dividers, these trabeculae. So it too is trabeculated. And it lies underneath your 9th, 10th, 11th rib. 9 to 11 rib. Why is that important? Well, in blunt abdominal trauma, if you see someone that has blunt abdominal trauma, maybe they were in a car accident, fell off a bike, etc., and now they're bleeding out, you're thinking of the spleen, because your spleen is very, very vascular. Or if they have some sort of fracture in their 9th, 10th, 11th rib, which will tell you on your question stem, and they're bleeding out, it's because their rib fractured, lacerated their spleen, now they're bleeding out. All right. So 9 to 11, if they show you that in the question stem, you should automatically think spleen laceration or rupture or anything. All right. That's kind of like their little code for spleen laceration. They won't just straight up say spleen laceration. Now your splenic artery and your splenic vein will connect to it at the hilum. What the heck is a hilum? A hilum is actually a botanical term. If you plant a seed, then a little sprout will come out of the seed, right? And that little area where the sprout comes out of the seed is called the hilum. So someone saw a spleen, saw these vessels coming out, kind of like a little sprout, kind of like a little tree, and said that's the hilum. All right? That's what it means by hilum. That they're not going to touch you on that. Don't try too hard to remember that. But that's why they call it your hilum. Okay. And does it have lymphatics? Yeah. Does it have lymphatic vessels? It does. It does not have afferent, which sends. It only has an efferent lymphatic vessel. What that means is that it only sends out lymphocytes and lymphatic fluid. It doesn't take in and kind of filter lymphatic fluids. It doesn't filter lymphatic fluids, then why are we talking about it here? Because it filters blood. Before we talk about how it filters blood, one more key fact, one more, I guess, trivia fact that they really like to test you on is that your spleen is found near your guts, and most of your foregut and its foregut organs, like your gallbladder, like your liver, come from your endoderm. Your spleen doesn't come from your endoderm. Your spleen comes from your mesoderm. They really like this question. I've gotten a question. They were talking about someone who fell off their bike, 9th rib fracture, they're bleeding out, and I'm thinking, okay, spleen laceration, I'm thinking all this pathology, and then all of a sudden the last sentence is, the organ that's damaged, what's its embryological origin? And you're just like, just a curveball, right? Just a curveball they throw at you. But now you know that your spleen comes from your mesoderm. Okay? That's enough about the little nitty-gritty anatomy facts. Let's talk about its function. How does it filter blood? Well, blood vessel will come in. Let me see if I have a red marker. Red for blood. Blood will come in. Oh, that's kind of chromy, but hopefully you can see it. Blood will come in. There it is. That's a little better. And immediately get greeted by follicles. What do your follicles contain? We just talked about it. It contains your B cells, don't they? And they make these little sheaths around your blood vessels. Make these sheaths around your blood vessels. And so the first contact your blood vessels come into is going to be your follicular B cells. And they kind of scan your blood, make sure there's nothing hiding in there. Okay? Around these little lymphoid sheaths is an area called your peri-lymphoid sheaths. Peri is kind of like para, which means around. So you have your peri-lymphoid sheaths. And they contain your T cells. So it's very similar to your lymph nodes, where fluid comes in and the first thing it contacts is your outside B cells. And the second thing it contacts is going to be your T cells. All right, why it does that, I don't know. Maybe your body figured out that's the best way to fight pathogens. Hit your B cells first, then hit your T cells. So your peri-lymphoid sheaths are the area outside, and they contain your T cells. Now, if you cut the spleen and you look at this area, it looks a little bit paler, looks a little bit whiter. We call this your white pulp. All right, so it shouldn't be too hard to remember that your white pulp contains all your white blood cells, your B cells, your T cells. Okay. As your blood vessels and your blood leaves this area, it will eventually drain into these highly vascular sinusoids, highly vascular holes. All right, so all your blood will drain here. And if you, again, cut the spleen and you look at this area, it looks very, very red, right? That's because all your blood's draining in there. So this we call the red pulp. The red pulp. And this has your sinuses that drain your blood. That's why it's so red. That's why it's so red. One thing they want you to know, in the red pulp you have these little cords, these cords of connective tissue. And your cords of connective tissue have macrophages in them. Just to pick up any stranglers that might be left, any pathogens that might have gotten through your white pulp. Okay. They sometimes call it splenic cords, sometimes they call it cords of Billroth. All you need to know is that there are some cords. And that is the anatomy of your spleen. Now, what happens if your spleen is damaged? What happens if your spleen is damaged? That is not a good thing. Your spleen helps filter your blood, but it also stores a lot of your blood. It actually stores a lot of other things too. Your spleen helps store your platelets, helps store your blood, actually stores up to about 30% of total blood volume, and stores your macrophages. In fact, it stores up to 50% of all your body's macrophages, so it stores a ton of stuff. And if you have some sort of splenic dysfunction, if your spleen's shot, then something can happen to your platelets. You can get thrombocytopenia. So that's platelets. How about blood? We already talked about it. It stores so much blood. If it gets lacerated, if it ruptures, you can bleed out like that. You can bleed out really, really quickly. So it stores your blood. Not only does it store your blood, but it filters your blood. It filters your blood, right? We talked about how it filters through your white pulp, through all these white blood cells, your B cells, your T cells. So it filters your blood. And your macrophages will eat any pathogens that might be in your blood, but will also eat any abnormal red blood cells. And they might be mutated or might be old or no longer works. Your macrophages will eat that too. So it will filter and destroy abnormal red blood cells. If you don't have a spleen, then you can't filter your red blood cells. You can't destroy abnormal red blood cells. So you'll see in your blood abnormal red blood cells. They survive. There's no spleen to get rid of them. You might see some red blood cells that still have their nucleus. If you recall, red blood cells don't have nucleus anymore, right? They're anucleated. We call these Howell Jolly bodies. You see this on a blood smear, then you look on the answer choices, look for some sort of splenic dysfunction. I've been asked this before. Or you might see a red blood cell that has a lot of membrane, too much membrane to do it any good. And the membrane kind of folds up and makes this little target. We call this target cells. Again, you see target cells, look at the answer choices, look for splenic dysfunction. All right, so your spleen filters and destroys abnormal red blood cells. You don't have a spleen, you won't be able to filter and destroy these abnormal blood cells. You'll see it in your blood. See it in your blood. So that's blood. Last thing is the macrophage part. Your spleen holds up to 50% of your macrophages, and your macrophages are needed to destroy pathogens, right? If you don't have a spleen, then you lose this giant reserve of macrophages, and it predisposes you to encapsulated bacteria. Encapsulated bacteria include things like H. influenza, includes things like strep, includes things like Neisseria, includes things like Salmonella. There are a ton more, but these are the big ones. Why is that? Why does losing your spleen predispose you to these bacteria? Well, your macrophages are really good at recognizing proteins on the outside. Well, encapsulated bacteria's capsules are made of proteins. They're made up of polysaccharides, sugars, carbohydrates. Macrophages can't recognize these. They can't eat them. They can't phagocytize them. But they really want to. So what does it do? Turn to your phagocyte buddies. That's your complements. That's your IGs, right? Complements. IGs. So these will coat the capsule, and your macrophages will say, now I can kind of eat them. Now I can kind of recognize them. Right? So in a normal person with a normal spleen, complements will coat these capsules, and your macrophages in your blood will eat them. And any that it missed will go to your spleen, and then your spleen has all these macrophages. 50% of your macrophages is waiting, and it sees them, kills them, all is good. If you lose your spleen, then you'll lose that, won't you? And so your complements and your IGs will coat all these bacteria, and some of it will get destroyed in the blood, but some will survive, and it'll go to your spleen, and it's going to your spleen, and it's thinking, oh no, all these macrophages are going to be there, we're going to be destroyed for sure. It goes to your spleen's shot, you have no macrophages, nothing happens. It leaves the spleen, goes back to your body, wreaks havoc. That is why encapsulated bacteria is so dangerous in people with splenic dysfunction. Understand? So they're predisposed to H. influenza, they're predisposed to streptococcus, Neisseria, meningitis, and Salmonella. The one they always like to ask is someone with sickle cell who lost their spleen and they have osteomyelitis. What do they have? Salmonella, osteomyelitis, okay? So you have someone without a spleen, predisposed to all these things. How can you help them? You want to help them. You're going to want to give vaccines for these. Give vaccines for H. flu, vaccines for streptococcus, vaccine for Neisseria, and that kind of reduces their chance of getting these diseases. Good? Cool. Lastly, I think the last one we're going to talk about is the thymus. Let's talk about the thymus. Your thymus is where your T cells mature. I didn't say where your T cells are made. I say where they mature. Your T cells are made in your bone marrow, but then they'll travel up to your thymus where they will mature into proper functioning T cells. Okay. Let's talk about your thymus. Your thymus is, surprise, surprise, encapsulated. So that is capsule. Seems like everything's encapsulated, right? And it's broken up into different regions. This first region, right under your capsule, is called your subcapsular region. What a great name. right under your capsule, we call it your subcapsular region. This outside region is going to call your cortex. You call everything your cortex if it's kind of on the outside, right? This inside region right here is your medulla, as are most inside regions. And then the one kind of in between called your corticomedullary. Okay. So your T cells are made in your bone marrow, and they will leave your bone marrow and travel through your blood vessels, through your venules, eventually make it to your thymus. So this is your little blood vessel, and your T cells will pop out. T cells will pop out. In fact, when I say T cells, I shouldn't actually call them T cells. They're not proper, full-grown T cells. They're actually, at this stage, thymocytes. They're immature, little snotty-nosed brats. They haven't matured and earned the right to be called T cells. So we call them thymocytes. So they leave your venules and they will migrate all the way up here. Migrate to the top subcapsular region. The top subcapsular region. And when they migrate up here, your T cells will sense that and they will start to rev up its genes. Especially the gene that makes T cell receptors. So they will start to express both CD4, and CD8. Express both CD4, CD8, both CD4, CD8. Now you know by now that they won't keep both CD4, CD8, but in the beginning they rev up their gene and they make both CD4 and CD8. All right, now they move down to the cortex, where epithelial cells will meet them. All right, so let's draw it back out. We have 4, 8, 4, 8, 4, 8. Your epithelial cells will meet them. And what they have is, and they want to test out these nifty new receptors. So what they have is MHC2, MHC1, MHC2, MHC1, MHC2, and 1. So they will test out these new receptors. And if 4 and 2 bind, rule of 8s, right? 4 and 2 bind, and they bind really, really well. They say, hey, you're binding really well. It's a perfect fit. You should keep that receptor. And says, hey, you're probably right. I'm going to keep that receptor. And the gene that was coding for CD8 will start to downregulate and die, and you will lose that receptor. So now you're just left with CD4. That becomes your CD4 cell. Makes perfect sense. How about hair? Well, if this binds CD8 and MHC1, and it binds really well, it's a perfect fit. And it says, hey, that's a perfect fit. You should keep that receptor for sure. It's a perfect fit. Keep that receptor for sure. And the cell says, you're absolutely right. I want to keep it. And so the gene that was coding for CD4 will be downregulated and eventually disappear. This becomes your CD8 cell. Easy peasy. How easy is that? So this is called positive selection. Positive selection. The things that fit perfectly, they get a survival signal and they survive to the next round. The things that bind too strongly won't get that survival signal. They'll die. Things that don't bind at all won't get that survival signal. They die. Most of the cells, most of your T cells actually die in this selection process. Most of them die here. But the ones that do make it through, let's draw it back out, CD4, CD8. We'll move on down to your corticomedullary and your medulla area, okay? Where it's tested again. But this time, it's not testing whether or not it will bind to MHC, MH1. We already did that. This time it's testing whether it will bind to self-antigens, your own body. And so self-antigens will get presented here. And see if these will bind to it. If it binds super, super strong to yourself, if it binds and attacks your own self-antigens, we don't want that. And so if it binds really strongly, it will get an apoptotic signal. apoptotic signal and die. If it doesn't bind that strongly, we say, okay, cool. It's not going to attack our own body. You passed all the tests. We're sending you on your way. Have fun. It becomes a fully mature T cell. This test is called your negative selection. And what it tests for is for autoimmunity. Making sure it doesn't bind too strongly and attack your own body. So we talked about positive selection up here, and then negative selection down here. One more trick question, or I guess a nitty-gritty factor they like to test you on is, it's kind of cool that your thymus makes these little antigens that are found in other parts of your body. So it might make some liver antigens or some splenic antigens, just to make sure your body won't attack your spleen, won't attack your own liver. It does this so it creates what we call tissue restricted antigens. It makes antigens that were supposed to be found in certain tissue areas only. Only found in your spleen, only found in your liver, where your thymus is able to make these because it needs to. Gotta make sure your T cells don't attack your spleen, attack your liver. So it makes these tissue restricted antigen. And this is controlled by the gene AIRE, also known as autoimmune regulator gene. Again, testing for autoimmune potential. A deficiency in this gene will not make these antigens, will not be able to test whether or not your cells will attack your own body, and will lead to autoimmune polyendocrine syndrome. You don't need to know too much about this other than the fact that because you're not able to test whether or not your T cells will attack your body, you get this autoimmune disease. Done with the lymphatic system and its associated organs. Thanks.